Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about bacterial conjugation. So this video is specially for +3 first year students because they have horizontal gene transfer in their syllabus in code 1. So let's start. At first, these are the modes of horizontal gene transfer: transformation, conjugation, transduction. In transformation, bacteria, those are competent, can uptake DNA from outside from another bacteria, but there is no physical contact between them, and only the competent cell can uptake the DNA inside it. Next, conjugation, the process which we will discuss in this video. In conjugation, there is physical contact between two bacteria through sex pilus of two bacterial cell. And transduction, where also there is no physical contact, but a bacteriophage is involved, which help to transfer the DNA from one bacterium to another bacterium. So basically these are three different modes of horizontal gene transfer in bacteria. Because they lack true sexual reproduction, they have parasexual reproduction, and these are three methods of this parasexual reproduction. At first, today we will discuss only about conjugation. It is the transfer of plasmid or other cell transmissible DNA element and sometimes chromosomal DNA from donor cell to a recipient cell. So what is donor and what is recipient? In case of bacteria, they do not have any male and female cell. So how do we recognize which is the donor cell and which is the recipient cell? So at first, the bacterial cell which have the F plasmid, they are called as the donor cell or F plus cell, because they have a particular type of plasmid. Apart from their normal chromosome or DNA, they have F plasmid, and they are regarded as F plus because they have F plasmid, and they can donate that plasmid to those bacterium those do not have that F plasmid, those lack F plasmid. Okay, so the bacterium having F plasmid is called as donor cell or F plus cell, and those lack F plasmid is called as F minus cell or recipient cell. Okay, and a recipient of the DNA transferred by conjugation, recipients of DNA transferred by conjugation, those who receive the DNA, that is F minus cell, can receive the DNA, and after conjugation, after receiving that DNA, they become transconjugants. This process of conjugation, can transfer DNA regions of 100 to thousands of kilobases of nucleotide, and has a broadest host range for DNA transfer among the methods for bacterial exchange, bacterial DNA exchange. The next, conjugation occur in and between many species of bacteria, including gram-negative and gram-positive. So broad host range conjugative plasmid have been used in molecular biology to introduce recombinant genes to bacterial species that are refractory to routine transformation and transduction. Okay, so we can modify that gene and also introduce it to the another bacteria. So we can do all those things. Although numerous examples of conjugative plasmid exist, conjugation involves a plasmid mostly. Okay. So what are the principle behind this process? The process of bacterial conjugation is based on the principle that the plasmid or any other genetic material is transferred from donor cell to the recipient cell through close physical contact between them. So in this process, physical contact is required. And how the physical contact is formed? Between the pilus, because pilus or pili is formed are present on the surface of bacteria, and those structures, apart from flagella, bacterial surface have pili and fibri. So those pili, which are hair-like projection, help in this conjugation process. And of all conjugative plasmids, a plasmid of E. coli was the first discovered and is one of the best studied. The A plasmid present in 1 or 2 copies per cell and is very large, containing about 100 kilobases. E. coli containing the A plasmid is called as F or male cell, A plus or male cell or donor cell, and E. coli lacking the A plasmid are referred to as recipient or female cell or F minus cell. Not only E. coli, all other bacterial cells those lack A plasmid regarded as recipient cell or H minus cell. The bacterial cell having A plasmid known as a donor cell or A plus cell or male cell. Only donor cell are capable of transferring the A plasmid to the recipient cell. For transfer of A plasmid from donor to recipient, intimate contact between cells occur, resulting in mating pair formation, and it is required in this process. Okay, so it is the principle behind bacterial conjugation. Now what are the steps involved? The donor bacterium extends a hair-like structure called pilus. The singular form of pili is the pilus. So one bacteria which act as donor, they extend their hair-like structure called pilus towards the recipient. Pilus attaches to the recipient bacterium and draws it in. A breeze is formed between these two bacteria which are involved. A breeze is formed. The donor bacterium transfers a copy of the plasmid DNA, to the recipient bacterium. Now, two bacteria which are involved in this process detach from each other, and that plasmid, after entering into the new bacterium, make a copy of it. Okay, and the recipient bacteria now become new donor and become the F plus. Okay, so it is the A plus with having the A plasmid, and those lack that F plasmid is called as F minus. So after entering into the body of F minus, the F plasmid, after entering into the F minus become F plus. Okay, in this way the transfer of plasmid occur between F plus and F minus, and F minus become F plus. So it is the overall process of conjugation. Now what is HFR? HFR means or high frequency recombination bacteria means these are the bacteria that have integrated conjugative plasmid. Like F factor gets integrated with their bacterial chromosome after entering into the cell of another bacteria. Sometimes this A plasmid gets integrated with the main bacterial DNA, and they become HFR or high frequency recombination bacteria. This allows them to transfer their chromosome during conjugation, because normally during conjugation only the F plasmid will transfer, but in case of HFR cell the bacterial chromosome can also transfer, because in those bacterial cell that F plasmid gets integrated with the normal bacterial chromosome. So it is about HFR cell. Now what is F minus cell? What is F dash bacteria or F dash bacterial cell? Bacteria that have F plasmid that also contains a portion of this bacterial genome. That means now HFR cells, sometimes the plasmid detach from the HFR cells. So it is the condition of HFR. Sometimes this plasmid gets detached again and get detached from the normal bacterial chromosome, and that condition is called as F dash cell. Those cells are called as F dash cell. So this is my handwritten note, and my handwriting is very bad, so please cooperate, just understand the things. At first, what is A plus and F minus? A plus, those are with F plasmid, and those lack F plasmid are called as F minus. So next is HFR, the case where the F plasmid gets integrated with the bacterial chromosome. Now what is F dash after HFR? What is F dash? So sometimes the disintegration occur where plasmid and chromosome gets disconnected from each other. Some part of chromosome remain connected with plasmid after disintegration. Some parts of chromosome sometimes accidentally remain connected with the plasmid, and rest of the chromosome remain free. Okay, so it is the F dash plasmid condition. Okay, so it is the overall F plus, F minus, HFR and F dash. So it is all about conjugation. I hope you get benefit from this video, you really enjoyed this video, and if you liked it, please hit the like button and please share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.